Now, next is fatty acid synthesis. So, as I have already told you, cholesterol synthesis, fatty acid synthesis, and ketone body synthesis, all these are like, the excess acetyl-CoA that is generated by glycolysis apart from the body needs will be converted to these compounds, and they get stored at times of need. They'll be reutilized. So, fatty acid synthesis also occurs in a well-fed state of the person. Okay, so it occurs in cytoplasm. Here, the input compounds are acetyl-CoA obtained from glycolysis and NADPH from HMP shunt. Okay, both are utilized for the fatty acid synthesis. So, first step is acetyl-CoA along with in the presence of NADPH from HMP shunt is getting converted into malonyl-CoA. Okay. That is a rate limiting step. See, in detail, each and every step and the intermediates are not necessary. Just an overall idea of fatty acid synthesis is sufficient. So, acetyl-CoA from glycolysis along with NADPH from HMP shunt come together and it gets converted into malonyl-CoA by the synthase enzyme. Okay, so that is a rate limiting step. So, by the acetyl-CoA carboxylase. So that synthetic enzyme here is acetyl-CoA carboxylase. That is a rate limiting enzyme. Okay, because it is adding a carbon dioxide group into it. So acetyl-CoA carboxylase is the rate limiting enzyme. This is the first step in fatty acid synthesis. And ultimately, this malonyl-CoA through a sequence of reactions get converted into the most commonly formed fatty acid in our body is palmitic acid. Okay, by the enzyme fatty acid synthase complex. So it's a complex enzyme wherein multiple, it's a multi-enzyme complex kind of thing. So multiple enzymes and multiple reactions occur to convert malonyl-CoA to the final product, that is palmitic acid. Okay, so that is what is fatty acid synthesis. Just take a screenshot of this, because in synopsis, this as such the schematic representation is not there, so you can take a screenshot of this page. Okay, fatty acid synthesis is quite simple. Apart from cholesterol synthesis, some of the acetyl-CoA from glycolysis, it in the presence of NADPH and by the enzyme acetyl-CoA carboxylase gets converted to malonyl-CoA. That is a rate limiting step. And this malonyl-CoA, through a complex of reaction by the multi-enzyme complex, fatty acid synthase complex to form palmitic acid. It's the most common fatty acid synthesized. Okay. Now, the fatty acids are produced and they are stored in our body, right? They are stored as the triglycerides or triacylglycerides. Fine. Now, these fatty acids during body needs, it should release energy, right? So that is how it occurs. That is where the oxidation of fats come. Okay, the fatty acid synthesized gets stored in our body, but at times of need, that is during fasting state, these free fatty acids will be released from the adipose tissue, and then that will initiate their beta oxidation. So their oxidation of fats can occur either, there are different types of oxidation, that is beta oxidation, alpha oxidation, omega oxidation, etc. But beta oxidation is the most common type of oxidation that occur in our body. So we have to learn about only that. Okay, the beta oxidation of fats was described by Franz Nutt. So during fasting state, due to the release of the free fatty acids stored in the adipose tissues into the blood, beta oxidation of fats will be triggered. Okay, so beta oxidation of fats usually occurs in 3 stages. First stage is the free fatty acid released from the adipose tissue, they should be activated. That is the first step. So first step is fatty acid activation. So this fatty acids, since we have already told the fatty acid synthesis and all those things occur in the cytoplasm. See, please remember, fatty acid synthesis occurs in cytoplasm. Okay, so the activation of fatty acids also occurs in the cytosol. Once they are activated, so what is meant by activation of fatty acid? That is, the fatty acid is getting converted to its acyl-CoA form. That is palmitic acid. Imagine the most common fatty acid synthesized is palmitic acid. So it is converted into its acyl-CoA form. That is palmitoyl-CoA. So similarly, other fatty acids means it's acyl-CoA forms. That's all. Okay. So that is just an example. By activation, it means the fatty acid is getting converted to its acyl-CoA forms. This activation as such occurs in the cytoplasm. Now, once this is activated, when the fatty acid is activated, then the next step is it gets exported to mitochondria by the carnitine shuttle, that is called as a carnitine carrier system. So the carrier molecule for exporting the fatty acids to mitochondria is carnitine carrier. Okay, that is called as carnitine shuttle. In the mitochondria, the oxidation proper occurs. That is the third stage of beta oxidation of fat, the third step. Okay, so here ultimately this will be converted into acyl-CoA and acetyl-CoA. See, how do we release energy? Even after glycolysis also, only pyruvate is formed, right? Then it gets converted to acetyl-CoA. This acetyl-CoA goes into citric acid cycle. So we learned citric acid cycle is the final common oxidative pathway for proteins, carbohydrates, and lipids, right? So after glucose glycolysis, acetyl-CoA formed entered citric acid cycle to release energy. Similarly, the beta oxidation of fats also release, ultimately the compound releases acetyl-CoA and acyl-CoA. So this acetyl-CoA goes to Krebs cycle or citric acid cycle to produce energy, whereas this acyl-CoA can re-enter this cycle. Okay, and when one palmitate molecule enters the beta oxidation, about 129 ATPs are produced. So a high amount of energy is produced as a result of beta oxidation of fats. Okay, you can take a screenshot of this slide. So beta oxidation occurs in 3 stages. When there is a fasting state, the free fatty acids stored in the adipose tissue will be released into the plasma. So within the cytoplasm, first these fatty acids get activated. That is, they get converted to their acyl-CoA forms. Example, palmitic acid getting converted into palmitoyl-CoA. Now, from the cytoplasm, they should move to mitochondria for the proper beta oxidation. So there, that exportation is brought about by the carnitine carrier system. Within the mitochondria, oxidation proper occurs. The final products will be acetyl-CoA re-entering Krebs cycle and acyl-CoA, which can re-enter this cycle again. So ultimately, at the end of beta oxidation of fat, 129 ATPs are generated from one molecule of palmitate. For other fatty acids, that differs. Okay, but since palmitic acid is the most common fatty acid which enters or which is produced as well as which enters the beta oxidation, we mention it as 129 ATPs. Is it clear? Okay. So, beta oxidation of fat, that cycle is quite important. Okay. So, when that cycle gets blocked, there are 2 important lethal complications associated with blockage of beta oxidation of fat. That is, one is sudden infant death syndrome and another is Jamaican vomiting sickness. Okay. So, you can expect questions from this session. Now, we'll discuss a few questions based on the fatty acid synthesis and beta oxidation. Okay, true regarding the reducing equivalent of fatty acid synthesis is. So in fatty acid synthesis, which is the reducing equivalent used? It is NADPH, so that is derived from HMP shell, and hence the answer is option C, both of the above. Transfer of fatty acid across the mitochondrial membrane is brought about by, yes, it is carnitine. Don't confuse between creatinine and carnitine. Okay. De novo synthesis of fatty acid occurs in. So de novo synthesis means just the fatty acid synthesis proper. It occurs in cytoplasm, while the beta oxidation proper occurs in mitochondria. Now, the conversion of acetyl-CoA to malenyl-CoA is a rate limiting step in fatty acid synthesis. Which of the following enzyme catalyzes the reaction? Is it acetyl-CoA carboxylase or acetyl-CoA decarboxylase? It is acetyl-CoA carboxylase. Okay. Where are the enzymes for beta oxidation present? So they have asked beta oxidation proper. So where are the enzymes for beta oxidation proper present? It is in the mitochondria. Very good. So beta oxidation of fatty acid occurs in all the tissues except. Here, the answer is all of the above. That is because beta oxidation proper occurs in mitochondria. Erythrocytes lack mitochondria, so their beta oxidation of fatty acid is not possible. Brain. Why can't brain utilize fatty acid? Because the free fatty acids cannot enter the blood-brain barrier. One thing is that, and another thing is, even though a few fatty acids that enter, they undergo alpha oxidation rather than beta oxidation. So beta oxidation of fatty acids cannot occur in brain. Then erythrocytes. Then also adrenal medulla also lack various enzymes for beta oxidation of fatty acids. So these are the three tissues where beta oxidation of fatty acid is not possible. Erythrocytes, brain, and adrenal medulla. Rest all tissues, beta oxidation of fatty acids occur. Okay. Erythrocytes lack mitochondria. Brain, fatty acid crossing the blood-brain barrier is difficult, and there mostly alpha oxidation occurs. Adrenal medulla lack a lot of enzymes involved in the beta oxidation. What is the net gain through the beta oxidation of palmitic acid? It is 129 ATPs, option C.